In this video tutorial, I am going to discuss about the mechanism of enzyme action. Now to understand about the mechanism of enzyme action, let's first consider one very simple model of enzyme catalyze reaction. We know that in enzyme catalyze reaction, enzyme binds with the substrate reversibly to form enzyme-substrate complex. This enzyme-substrate complex then further undergo reaction to give rise to free enzyme and free product. Now if we want to understand this full mechanism of enzyme action, we can divide it into two parts. The first part is that how enzyme-substrate complex is formed. So the first thing that we need to learn is about the mechanism of enzyme-substrate complex formation. And the second part that we need to learn is that what enzyme does to the substrate so that substrate is getting converted to the product and enzyme is released as a free and unchanged, right? So the second aspect that we need to learn is the mechanism of enzyme catalysis. Now before we discuss about both of this mechanism of enzyme substrate complex formation and enzyme catalysis, let's consider the energy changes in any chemical reaction. Now to understand these energy changes, let's consider one hypothetical example. In this hypothetical example, what happens? There is one diatomic molecule. Here it is A bonded with the B. This diatomic molecule AB, it reacts with the monoatomic molecule C. And in this chemical reaction, let's say the bond between A and B is broken and there is a new bond formation between B and C. So the resultant product will be free A and second product is going to be B and C, both are bound with each other, right? Now in this chemical reaction, you can easily imagine that while this reaction is going on, there must be some intermediate being formed in which the bond between A and B is on verge of breaking. So the bond between A and B is on the verge of breaking and the bond between B and C is on verge of forming, right? So such intermediate you can imagine. Such intermediates are always formed in any chemical reaction. These intermediates, they are highly unstable and they are called as a transition state. Transition state. Now let's see that how in this reaction energy changes occur. To understand that, we will draw one graph that on the y-axis we take a free energy and on the x-axis we will take a reaction coordinate. So on the y-axis we will take free energy and on the x-axis we will take a reaction coordinates, right? So if we look at the energy changes during this reaction, the graph will be like this, right? Now what happens? At the initially we have only substrate. So the free energy of substrate is represented by this part. So this is the free energy of this diatomic molecule AB and C. Whereas the free energy of the product is represented by this line. So here this is the free energy of products, that is A and BC molecule. The peak energy is represented by this transition state because transition state is the highly unstable and it has a maximum free energy. So this is the free energy of A, B and C. And in this transition state, as I told you earlier, the bond between A and B is on the verge of breaking and B and C is on the verge of forming, right? So as you can see, if you consider this reaction end to end, that is from the substrate to the product, then you can say that the free energy of the substrate is this much and the free energy of the product is this much. So this much energy is being decreased, or you can say this much energy is being changed during substrate to the product. So this particular difference in the free energy is called as a delta G. Remember, the free energy is called as a G. Okay, so the difference we will call it as a delta G, and this is the difference of free energy of entire reaction. So this will be called as a delta G reaction. Now what happens? In this reaction, this much energy is getting released. But initially, this reaction to happen, initially this much energy from the starting from the substrate's energy to the transition state, this much energy needs to get invested into this reaction, then only this reaction is possible, right? So this much energy first needs to get invested in the substrate. So this energy difference, it is called as a activation energy. And by the symbol, we can represent it by the delta G double dagger, right? So this is the activation energy. Now we can easily understand that the higher the activation energy, then there is a higher the barrier between the substrate and product. It will be easy to understand, right? If more energy's investment is required at this place, then there are less chances that substrate can get converted to the product, right? Now how enzyme appears into this picture? See what enzyme does. Enzyme simply brings down this activation energy, right? So this green line, it represents that if the same reaction is catalyzed by the enzyme, and what enzyme does? Enzyme brings down the activation energy. So the new graph, which is represented in the green line, it looks like this. So here, as you can see, when enzyme comes into the picture, it brings down this activation energy. It has brought down activation energy by this magnitude, right? So because now the less energy is invested, this reaction will become a faster, and that is how enzyme carries out its catalysis. But you need to understand one thing very, very clearly, that whether enzyme is present or not, this delta G of reaction does not change, okay? Whether enzymes are present or absent, delta G of reaction is not going to be altered or changed. So remember, this delta G of reaction is unchanged by enzyme. And as this delta G of reaction is unchanged, we can say that equilibrium between this substrate and this product remains unchanged. So the equilibrium does not change. Now what does it mean? Now to understand this equilibrium does not change by the enzyme, let's consider one example. Suppose there is one reaction where we have 100 molecules of the substrate, and without the enzyme, in absence of enzyme, this 100 substrates are getting converted to the product, but only let's say 25 substrate is able to get converted to the product, and the time required for this process to occur in the absence of enzyme is, let's say, 24 hours. Now in presence of enzyme, what happens? Substrate will be able to get converted to the product, but this velocity is going to be very, very fast. So let's say the time over here is only 1 minute. But although this reaction occurs fast, it will not change its equilibrium. That means 100 substrate will be able to give rise only 25 substrate, whether enzyme is there or not. So here also 100 substrate will be able to give 25 product. Only the difference the enzyme makes is on the time scale. So you can say that velocity is increased. So I think that energy changes in chemical reaction is clear to you. So now let's proceed to the mechanism of enzyme-substrate complex formation. Now there are total 3 mechanisms that we need to understand about the enzyme-substrate complex formation. The first mechanism is the Fischer's Templet theory, which is also called as a lock and key model. The second mechanism is the Koshland's induced fit theory. And the third mechanism is the substrate strain theory. Let's consider all these 3 mechanisms one by one. Now this Fischer's Templet theory, which is also called as a lock and key model, it was the first ever theory for the explanation of the enzyme-substrate complex molecule. It was proposed in the year 1894 by the German biochemist Emil Fischer. According to this theory, the structure of enzyme is rigid, and because of the rigid enzyme structure, its active site, this is the active site of enzyme. Its active site is a preformed template. Now, to this preformed template of the active site, the substrate which is complementary to this preformed template binds with the enzyme, and such enzyme-substrate complex is formed. So what happens? Only a specific substrate whose shape is complementary to the active site can only bind at the active site of enzyme, and so enzyme-substrate complex forms. There are several advantages of this Fischer's template theory. What are these advantages? This theory was very simple to understand. It was very intuitive also. This theory also laid a foundation for the enzyme-substrate complex formation. So it has a historical value. It kept historical foundation for this theory. And the third advantage is that here this theory, it explains the high specificity of the enzyme. As you can see, the preformed template is there on the enzyme, and to which only specific substrate can bind. So this theory, it explains high specificity of certain enzymes. Now there are certain limitations of this theory. The first limitation is that as enzyme is rigid and its active site is preformed, then we can say that enzyme is not a flexible, right? So this theory, it cannot explain about the allosteric modulators. We know that allosteric modulators are the molecule which influences the binding of substrate with the enzyme. And that influence can only be through a flexible nature of the enzyme. And as in this theory, it is rigid, this allosteric modulators effect cannot be explained by this Fischer's template theory. The second limitation is that as we had seen earlier in this case, that there is a provision of the transition state. In this theory, as you can see, the substrate is not changing over here after binding also. So this theory fails to explain the transition state of substrate. So this can also be not explained by this theory. Okay. Now to address this limitation, second theory was introduced by the scientist named as a Koshland, and this theory is the Koshland's induced fit theory. It was introduced in the year 1958. According to this theory, enzyme is not rigid, rather enzyme is flexible. And as you can see, its active site, this active site is not fully preformed, rather active site is a partially formed. So such partially formed active site, when it binds with its complementary substrate, what happens? Initially, loose binding between substrate and enzyme occurs. This loose binding introduces the conformational changes in this enzyme, and so because of this conformational change in the enzyme, the shape of active site changes, and shape of active site changes in such a way that it nicely fits this substrate into its active site, and so binding will become stronger over here. So there will be more tight binding between this enzyme and substrate, right? Now the good point about this theory is that as here enzyme is flexible, and so as per the conformational changes in this enzyme, the shape of active site can be changed. It can very nicely explain about the allosteric modulators. Okay, so effect of allosteric modulators can be explained by this theory, but there are certain limitations also. What is that limitation? Just like our previous theory of Fischer's template theory, here also, as you can see, substrate, the shape of substrate is not being changed over here. So this limitation is that the transition state of the substrate cannot be explained by this theory also. So to address this limitation, third theory was introduced, and that is the substrate strain theory. According to this theory, what happens? Enzymes are flexible. Its active site is not fully formed, rather active site is partially formed. So just like our previous theory, that is Koshland's induced fit theory, here also enzymes are flexible and active site is partially formed. To this active site, substrate binds. Initial substrate binding is a loose binding, and this loose binding introduces the conformational changes in the enzyme. Due to these conformational changes, the shape of active site changes, and so there is very nice binding between substrate and enzyme occurs. Now, this was the same as of the Koshland's induced fit theory. But here in the substrate strain theory, one thing is more, that is, while the binding will become tight, nice and fit, the substrate undergoes strain. Substrate changes its shape and it becomes highly unstable. So as you can see, this shape of the substrate, we can call it as a transition state, right? So this theory is the modern theory. It explains all the aspects of enzyme substrate binding. See, here enzyme is flexible. So the good point is it can explain allosteric modulators. And as the shape of substrate is also being changed, we can say that this theory can explain the transition state of substrate. So this completes our understanding about the first mechanism, that is mechanism of enzyme substrate complex formation. Now let us look at the mechanism of enzyme catalysis. So next we will understand about the mechanism of enzyme catalysis. We had already seen that how enzyme substrate complex forms, right? So let's say suppose this is the active site. This is the active site of the enzyme. And with this enzyme, this substrate is bound, right? This mechanism we had already seen. Now what we need to understand? We need to understand mechanism of enzyme catalysis, right? So now we will understand that what happens at this active site. And because of this process which happens at the active site, this substrate is able to get converted into the product. So here at the active site, there are mainly 4 processes occurs. These 4 processes, it may be only one process may be acting, or it may be multiple simultaneous processes may be occurring. So let's see what are these 4 processes. The first and most common process is, that is acid-base catalysis. The second one is the covalent catalysis. The third one is the proximity catalysis. And the fourth and last one is the substrate strain catalysis. Let's look at all these processes one by one. The first, acid-base catalysis. We know that amino acid, they have a R group. So at the active site of the enzyme, certain amino acid side chain, they act as a base as well as acid. One of the very good example is of the histidine. Histidine side chain at a physiological pH, it acts as a both. It acts as an acid and it also acts as a base. Other such amino acids are the tyrosine, then cysteine, right? So what happens? Such amino acid, by being present at active site, this acid and base effect will be carried on the substrate, and so substrate gets converted to the product. One very good example of such enzyme is the ribonuclease A. This ribonuclease A, it breaks phosphodiester bond of RNA molecule, and this bond breakage is carried out by the acid-base catalysis. We will not go into the fine chemical details how this exactly acid-base catalysis occur. But if you are really interested to understand this mechanism, then you can comment in this video. If there are enough comments, I will definitely make a video about the fine chemical reactions in this acid-base catalysis. The second is the covalent catalysis. See, we know that there are certain amino acid which has negative side chain and certain amino acid has a positive side chain. So what happens? This negative and positive charge on the amino acid side chain at the active site, it will make up a bond with the substrate, and so necessary changes is done in the substrate and substrate gets converted to the product. One very good example of such enzyme is the Serine proteases. This is actually not a single enzyme, rather it is very big group of enzyme. It is called as a serine protease because there is a presence of serine amino acid at the active site of this enzyme. In this group, as a group member, we have very good examples of a chymotrypsin, then trypsin, thrombin, etc. So this all enzyme, they breaks down a protein molecule. The third is very interesting. It is a proximity catalysis. See what happens whenever there are two or more substrate for any enzyme. So let's say this is the enzyme, this is the active site, and what happens as this enzyme has, let's say this enzyme has a two substrate, one substrate over here and second substrate over here. So what happens? By just presence of enzyme, these two substrate are brought near to each other. In absence of enzyme, what will happen? This both substrate will be very far from each other. But as enzyme has a binding site for both the substrate, what will happen? Both substrate comes near to each other. They become a proximal to each other. And because of the proximity to each other, the reaction between both of the substrate will become a feasible one. And so it can give rise to product. So this is how proximity catalysis works. The last one is the substrate strain catalysis, and this partially we had already seen in the third mechanism of enzyme substrate complex formation, and that is the substrate strain theory. So here, as you can see, the binding of substrate to this enzyme, what happens? There is a strain is imparted on this substrate, and because strain is imparted, there will be a reaction in this substrate and substrate will be able to get converted. So that is how this substrate strain catalysis occurs. The example of substrate strain catalysis is the lysozyme. This lysozyme, it breaks down beta 1,4 glycosidic bond. You need to remember one thing very clearly, that it is not that one enzyme, if it is working on acid-base catalysis, then it will not work on other type of catalysis. Most of the time it is observed that multiple of this process occurs simultaneously at the active site to bring about catalysis. So that's all about the mechanism of enzyme action. I hope that all the aspect is clear. If you still have any query or confusion, please write it down in the comment section below. Thank you.